Hey y'all! Today we'll be taking a look at a medical research paper from the Stanford Med School Department of Ophthalmology. This paper was published in February of 2019. I've linked the full paper and its authors in the description below. In this video, I'll break down the purpose, methods, and results of this paper in the next few minutes. Firstly, let's define the key terms used in this paper. Anterior ischemic optic neuropathy, or AION, is a condition in which reduced blood flow, called ischemia, damages the interior of the optic nerve. AION is the most common type of sudden optic nerve damage in white adults over 50. ER stress, or endoplasmic reticulum stress, is a cellular condition in which the ER has reached its capacity to fold proteins. Sometimes, short-term ER stress is okay, but prolonged ER stress is bad and can lead to programmed cell death. A key signal of ER stress is the unfolded protein response, or UPR, when unfolded proteins are being released by the ER. CCAAT enhancer binding homologous protein, abbreviated as CHOP, is a transcription factor that induces cell death as a result of prolonged ER stress. Binding immunoglobulin protein, referred to in this paper as GRP78, is a chaperone in the endoplasmic reticulum that helps newly synthesized proteins bind to the ER and fold. Finally, 4-phenylbutyric acid, or 4-PBA, is a therapeutic chemical chaperone that treats urea cycle disorders. Recent research suggests that 4-PBA assists with stabilizing ER proteins before they reach the cell's surface, significantly reducing ER stress. The purpose of this study is to demonstrate how AION causes ER stress in the retina and optic nerve by investigating key molecules that form, and to investigate how 4-PBA can be utilized in its treatment. Moving on to methods, this study utilizes adult male and female C57 black mice, kept at constant temperature, a 12-hour light dart cycle, with food and water available at all times. All animal handling was done in accordance with Stanford's panel on laboratory animal care. For actual experimentation, mice were sedated with a cocktail of ketamine and xylazine. Some mice's pupils were dilated with 1% tropicamide and 2.5% phenylephrine hydrochloride. In order to induce AION in a mouse, a researcher fired low-energy transpupillary lasers into one eye, leaving the other, contralateral eye, as control. Some mice were treated with 4-PBA while still alive, undergoing injections of 250 microliters every day for 19 days. Throughout the study, mice retinae were frequently measured for structural changes through optical coherence tomography, an imaging technique that uses light to capture high-resolution images in biological tissue. Ultimately, all mice were sacrificed for histological analysis via Western blot. Investigators found that transcription factor CHOP increased significantly in the optic nerve 1 day after AION was induced, suggesting that AION caused ER stress, ultimately leading to cell death in the retina and optic nerve as a result of CHOP expression. Investigators also found that GRP78 levels decreased in this 1-day period, but this result should be taken with a grain of salt due to the extraordinarily high p-value of 0.65 associated with this measurement. This high of a p-value indicates that there's a good chance that this measurement is not statistically significant. In the group of AION mice that received injections of 4-PBA, investigators found that retinal and optic nerve cells survived at significantly higher rates, suggesting that 4-PBA reduced ER stress and could be a possible treatment for AION. This paper concludes that ER stress reduction in the retina and optic nerve is key to treating AION's effects and should be considered in human treatment, via 4-PBA or otherwise. I barely scratched the surface of this study with this video. Please do read the paper in its entirety, which is located in the description below. Let me know if you have any feedback in the comments below, and as always, if you noticed any inaccuracies, please contact me ASAP. Thanks for watching.